Hello everyone. Today we will have a video on mitosis. Let's briefly discuss the interphase, which is where most of the cell cycle is spent. This is a time when a cell's metabolic activity is very high and the cell performs its various functions within the organism. Moreover, a cell in interphase increases its supply of proteins, creates more cytoplasmic organelles such as mitochondria and ribosomes, and grows in size. Additionally, the chromosomes duplicate during this period. Typically, interphase lasts for at least 90% of the total time required for the cell cycle. Interphase can be divided into three main categories: the G1 phase, the S phase, and the G2 phase, where G1 and G2 stand for first gap and second gap, or first growth and second growth. During all three subphases, the cell grows. However, chromosomes are duplicated only during the S phase. S stands for synthesis of DNA, also known as DNA replication, which we'll have another video on. At the beginning of the S phase, each chromosome is single. At the end of the phase, after the DNA replication, the chromosomes are double, each consisting of two sister chromatids. To summarize interphase, a cell grows in G1, continues to grow as it copies chromosomes in the S phase, and then grows more as it completes preparations for cell division in G2. Prophase is the first phase of metaphase. During prophase, changes occur in both the nucleus and the cytoplasm. Within the nucleus, the chromatin fibers become more tightly coiled and folded, forming discrete chromosomes that can be seen with the light microscope. The nucleoli disappear, and each duplicated chromosome appears as two identical sister chromatids joined together, with a narrow waist at the centromere. In the cytoplasm, the mitotic spindle begins to form, as microtubules rapidly grow out from the centrosomes, which begin to move away from each other. During metaphase, the second phase, the mitotic spindle is fully formed with its poles at opposite ends of the cell. The chromosomes convene on the metaphase plate, an imaginary plane equidistant between the two poles of the spindle. The centromeres of all the chromosomes are lined up on the metaphase plate. For each chromosome, the kinetochores of the two sister chromatids face opposite poles of the spindle. The microtubules attached to a particular chromatid all come from one pole of the spindle, and those attached to a sister chromatid come from the opposite pole. During anaphase, it begins when the two centromeres of each chromosome come apart, separating the sister chromatids. Once separate, each sister chromatid is considered a full-fledged daughter chromosome. Motor proteins of the kinetochores, powered by ATP, walk the daughter chromosome centromere first along the microtubules toward the opposite poles of the cell. As this happens, the spindle microtubules attached to the kinetochore shorten. However, the spindle microtubules not attached to the chromosomes lengthen. The poles are moved far apart, elongating the cell. Anaphase is over when equivalent and complete collections of chromosomes has reached the two poles of the cell. Telophase is roughly the reverse of prophase. The cell elongation that started in anaphase continues. The outer nuclei envelopes form around the chromosomes. Meanwhile, the chromatin fiber of each chromosome uncoils and nucleoli reappear. At the end of telophase, the mitotic spindle disappears. Mitosis, the equal division of one nucleus into two genetically identical daughter nuclei, is now finished. To finish off cell division, we have cytokinesis, occurring after mitosis, which is the division of the cytoplasm itself, usually occurring along with telophase, in which the two daughter cells completely separate soon after the end of mitosis. In animal cells, cytokinesis involves a cleavage furrow, which pinches the cell in two, whereas in plant cells, a cell plate is formed that eventually turns into a cell wall, separating the two cells. Well, I hope that was helpful for all those students out there wanting to learn more about mitosis. If you want to keep in touch with the videos and learn more about biology topics, please feel free to subscribe. Thank you.